Regulatory RNAs. There are two major functional classes of RNA, non-coding and coding. Non-coding RNAs or ncRNAs, in short, are the RNA molecules transcribed from genomes that does not encode proteins. Non-coding RNAs play a major role in epigenetic regulation of gene expression in addition to their roles at the transcriptional and post-transcriptional levels of gene expression. Recent studies have revealed that about 90% of the eukaryotic genome is transcribed. Interestingly, only 1 to 2% of these transcripts code for protein, and remaining majority are transcribed as ncRNAs or non-coding RNAs. Non-coding RNAs are of many types, namely two main types: infrastructural ncRNA and regulatory ncRNA. Infrastructural ncRNA and regulatory ncRNA. Infrastructural ncRNA seems to have a housekeeping role in translation and splicing. They include ribosomal RNA, transfer RNA, and small nuclear RNA. Regulatory ncRNAs are interesting in that they are involved in the modification of other RNAs. There are different kinds of regulatory ncRNAs, which are microRNAs, PV interacting RNAs, small interfering RNAs, long non-coding RNAs, enhancer RNAs, and promoter associated RNAs. So microRNAs, miRNAs, are small single-stranded molecules, 20 to 24 nt, that derived from hairpin-like transcripts called pre-miRNA. Such a transcript is processed into mature miRNA to form the RNA-induced silencing complex, RISC, which contains miRNA interacting proteins such as DICE. miRNA will pair with complementary sequences on target mRNA transcripts, leading to gene silencing of the target. Then PV interacting RNAs, piRNAs. piRNAs are small ncRNAs, 24 to 31 nucleotides, that are able to form complexes with PV proteins, PIWI. piRNAs are characterized by an uridine at the 5' end and 2-O-methyl modification at the 3' end. Their main role is in silencing of transposable elements during germline development. Then small interfering RNAs, siRNAs. Mature siRNAs are 20 to 24 nt long, linear and dsRNAs processed by DICE. They mediate post-transcriptional silencing by a process called RNA interference, RNAi, in which siRNA interfere with the expression of complementary nucleotide sequence. Then long non-coding RNAs, INC-RNAs. INC-RNAs are considered as non-protein coding transcripts greater than 200 nt in length. The majority of non-coding RNAs belong to this group. Many of the INC-RNAs are subjected to splicing, polyadenylation, and other post-transcriptional modification. INC-RNAs are involved in epigenetic gene silencing and tumor development. They promote the expression of the genes that are involved in metastasis. Then enhancer RNA and promoter-associated RNAs. These two types of ncRNAs have been described recently, and their role are still unclear. Enhancer RNAs are non-coding transcripts with an average of 800 nt, ranging between 0.1 and 9 kilobase pairs. So eRNAs, small e, enhancer, are believed to function as transcriptional activators. Then PAR are non-coding transcripts that range from 16 nt to 200 nt. Most of the PARs are associated with highly expressed genes, even though they are weakly expressed and have short half-lives. This is believed that PARs are associated with transcriptional activation and repression. RNA interference, RNAi. RNA interference is an evolutionary mechanism for silencing gene expression. By it, an mRNA is silenced, inhibiting the production of the protein coded by it. It is found in plants and animals, so it is believed to have been present in a common ancestor of both. RNAi might have evolved as a host defense mechanism against viruses and other foreign nucleic acids. It forms an innate and adaptive immune response against viruses in plants and animals. Viruses have already evolved countermeasures to abrogate the RNAi response. Then scientists have been working in this field from 1990 onwards. Finally, Andrew Fire and Craig C. Mello shared the Nobel Prize for Physiology and Medicine for their work on RNAi in the nematode worm Cenorhabditis elegans, which they published in 1998. RNA interference probably comes into play when a double-stranded RNA is introduced either naturally or artificially into a cell. An ribonuclease enzyme cleaves the long dsRNA into small pieces. The small pieces could be miRNAs when the long dsRNA is endogenous, or siRNA when the dsRNA is exogenous. These two types of RNAs can bind to specific mRNAs to complementary hydrogen bonding and thereby inhibit its translation. Then application of RNAi. Gene knockout. RNA interference is employed to study the physiological roles of various genes. This is achieved by silencing the targeted gene to produce knockout cells, knockout animals, etc. Then functional genomics. Functional genomics is a study of how genes and intergenic regions of genome contribute to different biological processes. By RNAi, the functional role of individual genes in plants and animals can be mapped. Then applications in medicine. The important application of RNAi include the control of the onset and progress of diseases, prevention of viral diseases, prevention of drug resistance, control of metastasis, prevention of neurological disorders, regulation of autoimmune diseases, etc. Then CRISPR-Cas9. In 1987, Japanese scientists at Osaka University noticed a strange pattern of DNA sequences in Escherichia coli. The gene had 5 short repeating segments of DNA separated by short non-repeating spacer DNA sequences. All the 5 repeating segments have identical sequences, 29 bases each, and each spacer sequence has 32 bases. The term CRISPR, or clustered separately interspaced short palindromic repeats, was coined in 2002 by a team of Dutch scientists led by Ruud Janssen. The CRISPR-associated genes, or Cas genes, code for enzymes that cut DNA. Cas9, or CRISPR-associated protein 9, is an enzyme that uses CRISPR sequence as a guide to recognize and cleave specific strands of DNA that are complementary to the CRISPR sequence. The CRISPR-Cas9 technique is one of a number of gene editing tools. Many favor the CRISPR-Cas9 technique because of its high degree of flexibility and accuracy in cutting and pasting DNA. In recognition of the importance of CRISPR-Cas9, Nobel Prize in Chemistry was awarded to Jennifer Doudna and Emmanuel Charpentier, I'm not sure about the pronunciation, in 7th October 2020. So these are short repeating spacer DNA sequences, or clustered regularly interspaced short palindromic repeats. Next, gene modulation or gene switching, regulation of gene action in development. The development of a unicellular zygote to a multicellular organism involves two genetically programmed processes, namely differential growth and cellular differentiation. This takes place precisely at the right time and exactly in the correct location in the embryo. Differential growth is a growth occurring at different rates and at different patterns in the different parts. Differentiation is a process by which cells get variously modified for different functions. Since all the cells of an adult organism are formed by the mitotic division of one and the same zygotic cell, they will have the same genetic makeup. The reason is that mitosis is an equational division which ensures that genes are equally distributed among the daughter cells, and also that genes are neither lost nor gained during it. Still, the adult cells are extremely diverse in form, structure, and function. This means that a single cellular genotype can give rise to many different cellular phenotypes. This is due to differential development of cells governed by differential action of genes. In differential gene action, genetic code or genes are believed to operate differently at different times and in different parts of the embryo. As a result, only the correct parts of the genetic code or the correct genes operate in the correct part at the right time. This is ensured by a mechanism known as gene switching or gene modulation. So, the development of a cellular zygote or to a multicellular organism involves two genetically programmed processes we mentioned earlier, like differential growth and cellular differentiation. These take place precisely at the right time and exactly in the correct location in the embryo. Differential growth is the growth occurring at different rates and in different patterns in different parts. Differentiation is the process by which cells get variously modified for different functions. Since all the cells of an adult organism are formed by the mitotic division of one and the same zygotic cell, they will have the same genetic makeup. The reason is that mitosis is an equatorial division which ensure that genes are equally distributed among the daughter cells, and also that genes are neither lost nor gained during it. Still, the adult cells are extremely diverse in form, structure, and function. This means that a single cellular genotype can give rise to many different cellular phenotypes, and this is due to differential development of cells governed by differential action of genes. So, in differential gene action, genetic code or genes are believed to operate differently at different times and in different parts of the body. As a result, only the correct parts of the genetic code or incorrect genes operate in the correct path and at the right time. This is ensured by a mechanism known as gene switching or gene modulation, which switches the appropriate genes on and off as and when required. It brings about the expression of only some genes and the simultaneous repression of others at a given time in a specific region. In other regions, some other genes may be expressed. At a later time, some of the previously repressed genes may get expressed and some of the previously expressed genes may get repressed. So the cells differentiated in different parts will be phenotypically different. This results in cellular diversity and the formation of different tissues. This differentiation and development are brought about by differential gene action through gene switching or gene modulation, in which genetic code is differentially used in different parts at different times. So differential gene action through gene switching occurs at three levels, namely differential duplication, differential transcription, and differential translation. Differential duplication involves the selective amplification of only some genes at a time. Similarly, in differential transcription, only some DNA genes undergo transcription, and in differential translation, only some of the mRNA genes gets translated or expressed. A typical example of gene switching is furnished by the formation of fetal and adult human hemoglobins. The protein part globin of fetal hemoglobin is formed of 2 alpha chains and 2 gamma chains, and that of adult hemoglobin is formed of 2 alpha chains and 2 beta chains. The synthesis of alpha chain is governed by the alpha gene and beta chain by beta gene, and that the gamma chain by the gamma gene. So it appears clear that alpha gene is functional throughout life, beta gene is presented in the embryo and expressed in the adult, and gamma gene is functional only on embryo and it gets switched off soon after birth. It has now been made clear that repression or switching off of genes in gene modulation is brought about by certain regulator molecules such as histones, basic proteins, and some hormones. Some other hormones and certain inducible substances can activate genes and thereby promote differentiation and development. Histones can bind with appropriate genes and repress their activity. The binding between the basic histones and the acidic genes DNA is very tight and hence the repression of genes. Only acidic proteins can lift this bonding and de-repress the gene.